point, if you're working on the lab, you can just set that aside. Feel free to keep it open because we're going to go back to that at the end. And when we read the results, we'll also have to do some taking turns a little bit there as well so that we can keep enough distance. So you can just set that aside. You'll have a chance to go back to that and kind of continue working on that later. But we are going to talk a little bit about how gel electrophoresis is used in the process of sequencing DNA and trying to actually read out that sequence of A's, T's, C's, and G's. And then we'll go back and talk about kind of what you're doing in this lab and what we're looking at. Today, we're just running a gel with food coloring because it's really easy to kind of visualize and see what's happening with food coloring. It's obvious. You can see it in the gel. And so it's a good way just to kind of practice and see how this technique works. Tomorrow, we're going to run a lab with actual samples of DNA, which works the same way, but the thing is DNA is not immediately visible. We need to do an extra step where we stain the gel to darken where the DNA is, and it's just a little trickier to look at. So today's like the practice run, and tomorrow we'll do the real DNA electrophoresis lab, okay? Okay, so yesterday as you started the pre-lab in the background, you were looking at the process and reading about this process called Sangar sequencing. This was kind of the first really widely used technique for sequencing DNA and reading out the code of A's, T's, C's, and G's in an organism's DNA. And the reason we're talking about it here is partly because it is a process that's still widely used, though they're developing newer and faster ways to sequence DNA all the time, but also because it builds on a lot of what you already know about DNA structure and DNA replication, and it uses this process of electrophoresis. So it kind of uses a lot of the things that we've already learned about DNA. So when we want to sequence a piece of DNA, so we have these little strands of DNA. Basically, they take the DNA from an organism and kind of chop it up into a bunch of pieces, and then they try and read it and reassemble the pieces. And we're going to talk about the basic procedure. We're not going to get into all the details. But in order to sequence a piece of DNA, they use the process of DNA replication. And so they throw these ingredients into a test tube. They have the DNA template strand on the left there. So that's the sequence that they're trying to read. That's what they're trying to figure out. They put in a primer. So they know the first couple letters of the sequence, and they put in an RNA primer. Because as you know from DNA replication, DNA polymerase can't just start building from scratch. It needs something to start. So you can see the primer sequence, that TGTT, is complementary to the bases down here at this end, TGTT, so that'll attach on. And then DNA polymerase will be able to continue adding bases to this 3' end. Oh, I fixed it this morning and it moved. It'll continue adding bases to that 3' end to continue growing the strand in that direction. I'm going to have to point and not draw today because that's obnoxious. So then, of course, we also need bases that polymerase is going to be able to add on there. So they put in a whole bunch of regular bases, but they also mix in some bases that have been fluorescently tagged in different colors. And so you can see in the picture here, in this case, the adenine, the base letter is the second to the last one there. The adenine is kind of purple, the cytosine is kind of red, the thymine is yellow, and the guanine is green. So they put in some fluorescently tagged bases, and those fluorescently tagged ones have also been slightly modified. You can see down at the bottom, they are missing an oxygen. And the reason that's important is that means as polymerase goes along, it's grabbing bases and adding in the complementary bases to kind of replicate the strand that we're trying to read. But if it grabs a fluorescently tagged base, it can't attach the next one because it's missing that oxygen. And remember, these attach to each other through dehydration reactions. And so if you're going to take out an H2O, you need an oxygen, right? And if the oxygen's not there, it can't attach another base, so it just stops. And so it copies this little sequence over and over and over and over. And you can think of it kind of this way. Imagine you have like a huge bin full of, I don't know, black marbles with some red ones thrown in there. And I tell you to pull out marbles until you get a red one, and then you have to stop. Sometimes you'd get a red one on the first one, right? And you'd stop. Sometimes you do it twice, and you'd get one on the second try. Sometimes you'd get one on, if you did it enough, eventually you'd get a red marble on the first try at least once, and the second try at least once, and the third. Does that make sense? Like eventually you'd get a red marble at every different possible point. That's what happens here. When we copy this enough, eventually, so if we look at this, we throw all this stuff into the test tube, and it just copies that little strand of template DNA over and over and over, and eventually we end up with strands that end, because as soon as we hit that fluorescently tagged one, it can't go any further. So we have some where it grabs the fluorescently tagged one first, and that's it. So we get these really little ones that have a fluorescently tagged base right here. And sometimes it gets it on the second try, and it stops here. And sometimes it gets it on the third try. And so eventually we get strands of all different lengths. And the last base is always one of those fluorescently tagged bases. You with me so far in how this works? Now we have all these strands of DNA of different lengths. And the last base is the fluorescently tagged one. So now we can use gel electrophoresis to separate all of these strands. And we've done this probably hundreds, maybe thousands of times. So there really wouldn't just be one of each, like in this picture. You'd probably have several of each length. Now, if we run these through a gel electrophoresis, remember electrophoresis separates based on charge, but also based on size. And so our shortest strands, the ones where it grabbed the fluorescent tag right away, they're going to travel the farthest. The next longest ones will travel the next farthest, and they will separate themselves out in the electrophoresis gel in this order. Does that make sense? So we use electrophoresis to separate them out. Then they actually use, because these were fluorescently tagged with different colors, they use a laser that reads the wavelengths of the colors to detect which fluorescent tag is at the end of each strand. So remember, if this is our electrophoresis on here, this one is working kind of vertically. So they started the sample up here, and I'm sorry already for how this drawing is going to work out. Oh, that wasn't too bad. They're moving down. So the ones down here you can see are the shortest strands. And the ones up at the top are the longest strands. So they can basically read, if they just move their laser from the bottom to the top, they're reading this genetic sequence from here to the next base to the next base. So essentially what they're doing is they're reading the complementary letters. This G went with this, or this C went with this G right here. And this G went with the C right above it. And so they're reading the complementary sequence as they go. Kind of make sense? I think it's kind of an ingenious technique, actually. And they get a readout that looks kind of like this on the bottom of the slide here, where they're actually recording the wavelengths of all those different colors of light. The important thing is that since each base is a different color, they can just go through and say purple, red, green, red, purple, purple, red. And that's the same as saying C, G, A, T, G, G, C, or whatever it might be. Questions? So that process uses what we know about DNA replication. It's using that same process. They have to put in a primer to get it started. And then DNA polymerase builds the complementary strand using those base pairing rules. But we also use gel electrophoresis. We randomly stop that replication at different points. So we get all these strands of different lengths. And then we use electrophoresis to separate them out according to length, so which basically puts the bases in order. So then we can go through and just kind of read those in order. Questions about that? Okay, so that's kind of the additional reading that went with your lab. Explains this again. So if you read that yesterday and it didn't make a lot of sense, you might be able to read through it today and hopefully it will make a little more sense. In the lab that we're doing today, like I said, we are not running DNA today, but gel electrophoresis can be used to sort out lots of different types of molecules. This is also frequently used for proteins. Today we're using it for the pigment molecules that we put into food colorings. And so the pictures you see up on the screen, you have some of these in your lab packet later, but these are actually the chemical structures of the molecules that make red food coloring red or green food coloring green. Some types of green food coloring, things like green and purple, sometimes it's an actual molecule that reflects as green to our eyes. Sometimes it's blue and yellow combined. And when you look at your electrophoresis in a little while, you'll be able to tell which one that type of food coloring is. But looking at these, take a minute and just turn to the person next to you and kind of decide, like, which of these would move the farthest through an electrophoresis gel and why? Which ones wouldn't move as far and why? Turn and talk about that just for a minute to kind of review how that works.
The bottom, okay, so how it's like 4 and then 3? Yeah. The one on the left side and the bottom right corner. That one.
No.
And then the one on
these
the
are
right of the way bottom.
There's 2 here, and 2 here, and 2 here, and 1 here.
So as you look at your results in a little bit, part of your analysis is going to be asking you, and I purposely didn't put the colors on here, because part of the results are going to be for you to figure out of the molecules that are in your lab packet, which one is the red, which one is the yellow, which one is the blue, based on what you learned from your electrophoresis, based on how far they traveled through the gel. You should be able to tell which ones are the smallest and which ones are the largest molecules. Okay? And if you're not 100% sure, the explanations in your lab is really the part I'm going to be looking at to make sure you understand how electrophoresis works. Not so much whether you totally got it right, but whether your reasoning is accurate in terms of how electrophoresis works. Does that make sense? So in a few minutes, we're probably going to give your gels like 5 more minutes or so. I'll go take a look at them in just a second here. But we're going to be going back to take a look at these in just a little bit. Yeah, okay, we've got like 25 more minutes, so we're good. When we go back, we'll do the same thing. We'll go in shifts. First of all, make sure the power source is off before you do anything else. Secondly, we're running 125 volts of electricity through those things right now. So it's important to have them off before you open it. But also, once you open it, the liquid in there is going to be kind of warm. It's not hot enough to burn you, but just if you reach in there to actually touch your gel, which you can, it's warm because you're running electricity through it and it heated it up. So just so you're not surprised by that, you will take the cover off, but we're going to try and leave the gels in there to measure if we can. If we have to take them out, that's fine. But we're actually going to reuse these gels for 5th hour because food coloring, you can just keep running it and it'll eventually migrate all the way out of the gel and we can reuse it. So you're going to be measuring how far each different pigment migrated from where it started. So remember, as things travel down a gel, they kind of stay in their lane. So like it's moving straight down from the well that you put it in. And you're going to measure from that well to kind of the center. Food coloring doesn't make really nice solid bars. It's just kind of like this blurry spot of color that you'll see. And you're going to measure how far it went. It might be easier to see if you put a piece of white paper underneath it so you can see the colors a little better, so you can try that and just see what gives you the best view. I would also recommend taking a picture of your gel so that you just kind of have a visual of what it looked like, okay? If we were doing this on paper, I'd have you sketch it, but we're not. We're just doing it on computer, so you don't need to sketch it, but you do need to kind of remember what that looked like as you analyze your results. And there's a data table where you'll record those distances. You only need to measure the 4 that you put in. So if you shared your gel with another group, you can look at theirs if you want to. You should have gotten the same results on both sides, but you only need to measure yours. Does that make sense? After you've measured yours, then you have a seat and you can work on the post-lab stuff, and the other group can go up and make some observations and measure theirs. And then, like I said, once they're done, we're actually going to plug them back in and keep them running and see if we can run that food coloring all the way off the end of the gel so I can reuse them again next hour. Questions? Okay. Give me a few minutes to kind of check on how these gels are going. And like I said, we'll probably run them another 5 minutes or so you can go back to working on your pre-lab. If there were parts of the Sangar sequencing stuff you didn't get, feel free to go back and answer those questions again. Otherwise, I will let you know when it's time to go start looking at results. People at home, you can please stick around because we are going to have you look at the results as well. But you can also pull up your lab off of Classroom and work on that for a few minutes until it's time to look at those results. So just hang around for a couple minutes and we will bring you back over to a lab table so you can gather some data in a little bit.
Oh, it's our goal to look at the separation to samples and